of enzymes are comparable to the methods used for the manufacture of other industrial products. The salient features are described, briefly described. So the salient features are the selection of the microorganisms, formulation of the medium, cultural medium, right? And the production process and recovery and the purification of enzymes. And this is our enzyme production. Industrial microbial production of enzyme processes steps we can categorize so there is this outline, right? So this is a very general outline. So first market there is this preserved inoculum, right? So preserved inoculum we have developed, we have then this step process we have generated this preserved inoculum, and then we have this inoculum development. We'll develop this inoculum minute quantities, right? So minute quantities we develop this inoculum, and then there is this inoculation tank, right? And then finally there is this fermenter, right? There is this fermenter tank, and then there is this cell disruption, fermentation certain time spent, right? All those microorganisms. There is this fermentation, and then there is the cell disruption, filtration, cell debris removal, and then there is this removal of nucleic acids, salt treatment, cool storage, filtration, final chromatography, final purification chromatography, freeze drying, and there is our enzyme powder. So this is the outline of the flow chart for the production of enzymes by microorganisms. Right, एकदमै general, एकदमै simpleton, extremely simpleton diagram. So we'll try to break down this process, right? So the selection of microorganism, the most important criteria for selecting the microorganisms are that the organism must produce the maximum quantities of desired enzyme in a short time, while the amount of other metabolites produced are minimal. So this is the major criteria, right? So once the organism is selected, strain improvement for optimizing the enzyme production can be done by appropriate methods, mutagens and UV arrays, right? I've already discussed about these methods. We have already discussed about media optimization ko barema. We have already discussed, and strain improvement ko barema pani we have already discussed, right? So first, there is the need for optimizing the strain, strain media optimization, strain optimization, media optimization. So for the chosen organism chosen, the inoculum can be prepared in the liquid medium. So there is this type there is this fermentation, submerged fermentations, there is solid state fermentations. So normally we study about the liquid medium or the submerged fermentation. So next slides we'll study about those kinds of fermentation, submerged fermentation or the solid state fermentation. So the formulation of the medium, the cultural medium chosen should contain all the nutrients to support adequate growth of the microorganism that will ultimately result in good quantities of the enzyme production. The ingredients of the medium should be readily available at the low cost and are nutritionally safe, right? These are some of the requirements. Some of the commonly used substrate for the medium are starch, hydrolyzed molasses, corn steep liquor, yeast extract, whey, soybean meal, some cereal, wheat and pulses, peanuts have also been used. So these are some of the common substrate that are used. Like the biomass, these are nothing but biomass. Fermentation simple. Fermentation is quite simple. Fermentation substrate simple, right? So they only require biomass. The biomass convert to complex enzymes or secondary metabolite. So this is one of the main fermentation. Interest as a major concern because the substrate are extremely common. right? These are extremely common substrate like starch, hydrolyzate, molasses. These are extremely common substrate, and from these common substrate, you can generate expensive products, right? So the pH of the medium should be kept optimal for good microbial growth and enzyme production. So these are some of the factors that should be controlled or checked. So the production process, the industrial production of enzyme is mostly carried out by the submerged liquid conditions and to lesser extent by the solid state fermentation. So there is these two paradigm, right? There are these submerged liquid conditions, right? And then there is this lesser extent by use bhairaku, but there is now this growing interest in solid state fermentation or the solid substrate fermentation, right? So तपाईहरु पढिसक्नु भएको छैन यो? Have you studied about the solid state fermentation and submerged fermentation? Are these terms new to you?
डेफिनेसन पढिसकेको.
डेफिनेसन मात्र पढेको छ, यसमा डिटेलमा पढेको छैन.
डिटेलमा थिएन.
ओके ओके, अब ठिक छ. अब म एकैछिनपछि यु स्टडी अबाउट दिस फर्मेन्टेसन टेक्निक, र हामीले चाहिँ विल स्टडी अबाउट अराउन्ड 10-12 methods to industrial methods of producing enzymes. त्यसमा पनि we'll try to see this process, है त. So this submerged fermentation मा चाहिँ लिक्विड हुन्छ, राइट? there is this लिक्विडमा हामीले ग्रो गरेर आउँछौँ त्यो सेल्सहरुलाई, अनि त्यो लिक्विड नै हुन्छ. सो तपाईहरुले एकदमै इजिली इमेजिन गर्न सक्ने, भिजुअलाइज गर्न सक्ने भनेको चाहिँ दिस इज दिस सबमर्ज्ड फर्मेन्टेसन हो, राइट? लिक्विड फर्मेन्टेसन. And then there is another kind of fermentation called solid state or solid substrate fermentation. That is something त्यति कमन चाहिँ हुँदैन यो, बट अब यसले अब दिस इज समथिङ, अब यसमा चाहिँ धेरै ग्रेट इन्ट्रेस्ट छ, है त. अहिले चाहिँ लट अफ इन्ट्रेस्ट हेज बिन इन सोलिड सबस्ट्रेट फर्मेन्टेसन. So in submerged culture type, the yield are more and the chances of infection are less. Hence, this is the preferred method. However, solid state fermentation is historically important and still used for the production of the fungal enzymes, amylases, cellulases, proteases, and pectinases. So solid state fermentation, solid substrate fermentation, traditionally चाहिँ एकदमै युज हुन्थ्यो. अहिले आजकल खासै धेरै युज हुँदैन, तर अहिले फेरि there is this renewed interest in solid substrate fermentation. Right? There is this extremely renewed interest in. अब अहिले most common भनेको चाहिँ this यो submerged liquid fermentation हो. एकदमै common, इन्डस्ट्रीहरुमा एकदम commonly used हुने भनेको. The medium can be sterilized by employing batch or continuous sterilization techniques. The fermentation is started by inoculating the medium. The growth conditions, pH, temperature, oxygen supply, nutrient addition are maintained at optimal levels. The froth formation can be minimized by adding anti-foam agents. So your batch and continuous process bioreactor. Have you studied about bioreactors? Bioreactor पढ्नु भएको छ छैन?
त्यो चाहिँ डिटेलमा पढेको.
Okay, thank you. That will be very easy for me. At least you have studied about the bioreactor. किनभने bioreactor को बारेमा चाहिँ I don't need to explain it. So there is this batch. There are these two methods, right? Two fundamentally different methods. It was a batch work. The first batch, second batch, third batch, or there is this continuous process sterilization. So batch is the more preferred way. Most industry मा पनि हुने भनेको batch नै process नै हुन्छ. So batch मा चाहिँ हामी we can start fermentation. The fermentation हामी start गर्न सक्छौं. Okay, and so all these levels, there is this bioreactor. We have already studied about this bioreactor. Bioreactor भनेको के हो? Anybody? Somebody define me so that हामी एउटै च्याप्टरमा छौं भन्ने कुरा थाहा हुन्छ मलाई. What is a bioreactor?
कन्ट्रोल्ड भेसल हो जसमा pH हरु सबै कन्ट्रोल टेम्परेचर, pH अनि अक्सिजनहरू
Okay,
सबै
yeah.
मिलाएर
Uh,
दिँदा हुन्छ जाप फर्मेन्टेसन अलाउ गरिन्छ
okay, yes. अब यहाँ चाहिँ this is a specialized kind of vessel, right? जसमा चाहिँ हामीले के गर्छौँ भन्दाखेरि एउटा डिजायर्ड जुन बायो ट्रान्सफर्मेसन छ त्यो क्यारी आउट गरिरहेको हुन्छौँ, राइट? सो हामी एउटा चाहिँ डिजायर्ड बायो ट्रान्सफर्मेसन गर्न खोजिरहेको छौँ। अब सकेसम्म द्याट ट्रान्सफर्मेसन चाहिँ हामी के गर्न खोज्छौँ, त्यसलाई चाहिँ एउटा स्पेसिफिक कन्डिसनमा एउटा स्पेसिफिक मेटाबोलिक पाथवेमा त्यसलाई गाइड गर्न खोज्छौँ। सो एउटा चाहिँ डिजायर्ड केमिकल ट्रान्सफर्मेसन के गर्न खोज्छौँ भन्दाखेरि डिजायर्ड इन्भाइरोमेन्ट एउटा कन्ट्रोल्ड इन्भाइरोमेन्टमा चाहिँ हामी विल ट्राई टु convert that desired transformation in a controlled environment. So the production of enzymes with the bioreactor, so that everybody is clear about bioreactor. So since there is a bioreactor, like they be complicated. So bioreactor, there can be different kinds of variables, parameters, all those variables and parameters, they have to be identified. What are the different factors, variables that influence the bioreactor environment? That can be pH or temperature or the oxygen supply, nutrient addition, and foaming. So there can be either maintain how the temperature should be maintained, how the oxygen supply, carbon dioxide supply, foaming, right? All those metabolite production, unnecessary or the undesired metabolite production, all these factors, there can be many factors, right? So different detail you want to control the environment, that depends upon that. So all these they are controlled. The production of enzyme is mostly carried out by the batch fermentation and to a lesser extent by the continuous process. So you have to be clear. Try conducting your experiment in batch fermentation. That is more easy. So continuous process is quite difficult to maintain that conditions are maintain difficult. Batch it's easy to work. So bioreactor system must be maintained sterile throughout the fermentation process. Right? Sterile sterile what happens? To undesired biotransformation and that will create downstream processing, it will create a havoc. So it has to be maintained sterile throughout the fermentation process. So normally the fermentation process, it can take a variable amount of time, 2 to 7 days, right? So there can be some desired enzymes produced during the time and other several non-desired metabolites are also produced, and we have to be able to recover and purify the desired enzyme. So this is about the production process. So how do we recovery and the purification of the enzymes? How do we do that? So the desired enzyme produced may be excreted into the culture medium, so that is the extracellular enzymes, or may be present within the cell, that is our intracellular enzymes. So all these factors, you say you have to define factors हुनुपर्छ है त जुन हामीले प्रोडक्सन गर्न आँटेको एन्जाइम चाहिँ इन्ट्रासेलुलर हो कि एक्स्ट्रासेलुलर हो इफ इट्स एन इन्ट्रासेलुलर एन्जाइम्स के हुन्छ द पोस्ट प्रोसेसिङ क्यान बी लिटल एक्सपेन्सिभ एन्ड लेन्दियर राइट सो पहिले नै डिजाइन गर्दाखेरि नै कस्तो किसिमको एन्जाइम जेनेरेट गर्ने हो है त्यसलाई चाहिँ रिकम्बिनेन्ट टेक्निकबाट बनाउने हो सो द्याट एक्स्ट्रासेलुलर त्यसलाई चाहिँ एक्सक्रिट गर्न सकोस् त्यो स्टुडेन्टले अल द्याट फ्याक्टर द्याट हेज टु बी determined and earlier, right? Depending on the requirement, and the commercial enzyme may be crude or highly purified. So further, it may be in the solid or the liquid form. The steps involved in downstream processing, that is recovery and purification steps included, will depend on the nature of the enzyme and the degree of the purity desired. So the downstream processing, it all depends upon the nature of the enzyme. If the enzyme is extremely thermolabile, you cannot perform certain experiments. So chromatographic column react if certain enzymes, if they react with the chromatographic column, that process they have to be excluded because that depends upon the nature of the enzyme and also on the what is the level of purity that is desired. So in general, the recovery of the extracellular enzyme which is present in the broth is relatively simpler compared to the intracellular enzyme. For the release of the intracellular enzymes, special techniques are needed for cell disruption, Microbial cell can be broken down by the physical means, sonication, high pressure, and glass beads. The cell walls of the bacteria can be lysed by the enzyme lysozyme, right? The enzyme. So for each, the enzyme beta glucanase is used. However, enzymatic methods are expensive. So there are many steps that your intracellular enzyme that will be add the complexity add गर्छ. So there are different steps methods. So there are these physical or the mechanical steps. There are these chemical steps by enzymatic steps. So lyso enzymes use गर्न सकिन्छ or the beta glucanase enzymes use गर्न सकिन्छ. So the recovery and the purification steps will be the same for the intracellular and the extracellular enzymes once the cells are disrupted and the intracellular enzymes are released. So the most important consideration is to minimize the loss of desired enzyme activity. So एकदमै major concern, major focus of the research to industrial research को चाहिँ focus के हुनुपर्छ भन्दाखेरि to minimize the loss of the enzyme activity. Like that. So जहिले पनि if you are intervening है त कुनै सिस्टम छ कुनै एउटा चाहिँ entity छ एउटा चाहिँ enzyme छ whatever that be त्यसलाई चाहिँ if you are purify गर्नु भनेको चाहिँ you are disturbing it, right? The disturb गरिरहनु भएको छ. So if you are disturbing it त्यसले के गर्न सक्छ, it can affect the enzyme activity. त्यसले चाहिँ enzyme activity लाई चाहिँ affect गर्न सक्छ. So you should be very careful. So while you are performing any activity, purification activity, recovery activity, so you should be very clear what are its impact on the enzyme activity. And there is this removal of the cell debris. Centrifugation or the filtration can be used to remove the cell debris. And there is this removal of the nucleic acids. So even these are the undesired items, right? Undesired components. Nucleic acids interfere with the recovery and the purification of enzymes. They can be precipitated and removed by adding the polycations such as polyamines, streptomycin, and polyethylenimine. So all these either chemicals, they can be precipitated and removed. Nucleic acids. Similarly, there is also this enzyme precipitation, right? Desired enzymes also, they can be precipitated by using salts, right? Ammonium sulfate precipitation or organic solvents, or isopropanol, ethanol, or acetone. Precipitation is advantageous since the precipitated enzyme can be dissolved in the minimum volume to concentrate the enzyme. So precipitation is very easy to concentrate the enzyme. So other techniques are liquid partition for the concentration of the desired enzyme can be achieved by liquid extraction using polyethylene glycol or polyamines. So there is this the differential solubility, right? So differential solubility what you can do is, so contaminants different nature liquid so they can separate, either segregate, either partitioning. So we can purify our desired enzymes. Concentration pure. So it can be used. So you polyethylene glycol or polyamines or use this can be done. So and then we can further purification ultra grade purification you require the help of the chromatography, and this includes ion exchange, size exclusion affinity, hydrophobic interaction, and dye ligand chromatography. So among these, ion exchange chromatography is most commonly used for enzyme purification, right? Ion exchange chromatography, more or less. Why? Because ion exchange chromatography is relatively less expensive compared to other chromatographic methods. So finally, these need to be dried and packed. So packing, the transportation, it depends upon the cost, how valuable your yield is. The enzyme of interest, how valuable that is. So this depending on. So the concentrated form of the enzyme can be obtained by drying. This can be done by the film evaporators or the freeze dryers, lyophilizers. The dried enzyme can be packed and marketed. For certain enzymes, stability can be achieved by keeping them in ammonium sulfate suspensions. So all these enzymes used for food or medical treatment must be of high grade purity and must meet the required specifications by the regulatory bodies. These enzymes should be totally free from the toxic materials, harmful microorganisms, and should not cause allergic reactions. So, in case of high grade purity Chinese therapeutic agents, there is this next the quality control. There is this next step whereby they should be free from all kind of toxic materials and harmful microorganisms. And so you take them in general methods, general overview too. So we'll try to study. Let's say हामी के गर्छौं अरू च्याप्टरहरु पढ्छौं है त So let's study. अब हामी there was the brief introduction मात्र थियो है त हामी त्यसको बारेमा डिटेलमा पढ्छौं है त पछि भन्दाखेरि while we are studying about different enzymes जुनलाई चाहिँ हामी industrially manufacturing गर्दाखेरि हेर्छौं. So that was the brief introduction. है त brief introduction मात्र गरेको थियौं. स्क्रिनमा के देखिरहेछ स्क्रिनमा के देखिरहनु भएको छ सबैजनाले ओके So solid and submerged state fermentation. These are two of the major ways in which we generate fermented products. So solid state fermentation, SSF, is a fermentation method used by several industries like the pharmaceuticals, food, textile, etc. to produce metabolites of microorganisms using solid support in place of the liquid medium. So generally, while you are trying to visualize a bioreactor system, so general tendency के हुन्छ भन्दाखेरि you visualize a big vessel, like mostly of stainless steel vessel. The stainless steel vessel मा चाहिँ you pour some medium that is liquid, and then there is this impeller छ, and then there are different equipments that can regulate temperature, oxygen, and different pH, right? All these things that need to be monitored. And then you visualize that way. The solid state fermentation is there is nothing like the liquid state. So instead, there is the solid support in place of the liquid medium. So it is defined as the growth of microbe without the free-flowing aqueous phase. Free-flowing aqueous phase, aqueous phase, but there is no free-flowing either. So please be careful either. So there is not this free-flowing aqueous phase. So SSF is alternative to submerged fermentation for the production of value-added products like antibiotics, single-cell protein, PUFAs, right? Polyunsaturated fats, enzymes, organic acids, biopesticides, biofuels, and aroma production. So the support used is generally grain brands, de-oiled oil seed cakes, and other substances like, right? Normally, brands, right? So, grain go external covering, right? So initially, mostly fungi were used in such fermentation, such as these microorganisms were considered to be very optimally active in very low water activity. Later, many microbial species and yeast were used to carry out such fermentation also. So initially चाहिँ, this method of production, solid state fermentation, it was only limited to the fungi. Fungi, since they can grow in very low water activity. Fungi, I think fungi they can I think even grow on honey. हनीमा पनि ग्रो हुन सक्छ फन्जाई तपाईं देख्नु भएको छ त्यो फन्जाईहरू हनीमा ग्रो भएको have you seen fungi growing on honey yeah के भन्नुभयो मैले बुझ्दै बुझिनँ so मैले के भन्न खोजेको भन्दाखेरि fungi initially they were used in solid state fermentation because this it can remain active even in or they can be optimally active in very low water activity. Generally bacteria like they need quite a lot of water. Water they need lots and lots of water to maintain that biological activity. So so there are many progress made in solid state fermentation. We are trying to shift towards solid state fermentation. I'll explain the So the microbiological process of solid state fermentation has generated greater interest in recent years because it can be used for a variety of purposes, supported by some authors who have even indicated numerous advantages over their liquid counterparts, submerged fermentation. So there has been this renewed interest in solid state fermentation. So what are some of the substrate used in solid state fermentation? It can be the sugar cane, bagasse, tea waste, right, and wheat bran, sawdust, apple pomace, and the coconut oil cake. So this can be the substrate used, right? So अघि नै भनिसकेको छु है त फर्मेन्टेसनको एउटा ब्युटिफुल पार्ट के हो भन्दाखेरि low cost material can be used as a substrate. Low cost material जुन हाम्रो substrate लाई हामी product मा convert गर्दैछौँ, so you don't need to have a एकदम expensive substrate चाहिँदैन. त्यो cell जुन छ जुन fermenter मा जुन एउटा चाहिँ we are using that cell, right? It can be bacteria, fungi or yeast. त्यो cell ले fungi ले के गर्छ भन्दाखेरि त्यो high value yield भएको चाहिँ material, right, enzyme or the secondary metabolite, it can generate those enzymes and secondary metabolite by consuming these sugar cane, bagasse, tea waste, wheat bran. These are extremely cheap, not so expensive materials. That's why there is this growing interest. The fermentation by so much interest and major it doesn't use very expensive raw materials. So there are many biotechnological processes that involve the growth of organisms in solid substrates in absence or near absence of the free water. So there is no free water, right? The only water that is absolutely essential. Submerged fermentation, there is lots and lots of water. To mediumly equation medium, there is this water rheological properties, even that you have to consider. Either viscosity is the property that you have to consider. So in solid state fermentation, there is never that case because there is near absence of water. So in solid state fermentation deals with substances that are solid and contain low moisture levels. The most regularly used solid substrates are cereal grains, rice, wheat, barley, and corn, legume seeds, wheat bran, cellulose materials such as straw, sawdust, or wood shavings, and wild range of plants and animal material. So essentially we are using biomass, right? Dried or semi-dried biomass. So most of these compounds are polymeric molecules, insoluble or sparingly soluble in water, but most are cheap and easily obtainable and present a concentrated source of nutrients for the microbial growth. So these are cheap but easily obtainable, and they are the concentrated source of nutrient for the microbial growth. So also there is renewed interest, but still lots and lots of industries still we use submerged fermentation. Submerged fermentation, what are the benefits of submerged fermentation? That's why why it is widespread भन्दाखेरि एकदमै कन्ट्रोल गर्न चाहिँ सजिलो हुन्छ है त. सोलिड स्टेट फर्मेन्टेसन इट इज भेरी डिफिकल्ट टु कन्ट्रोल. जुन त्यो सोलिड स्टेट while you are conducting solid state fermentation, जुन त्यो environment छ environmental control गर्न चाहिँ it's quite difficult. So solid state fermentation can be defined in terms of the following properties, right? What are the different properties solid state fermentation defined गर्नको लागि? So porous matrix, porous matrix can be biodegradable or not, but the large surface area per unit volume in the range of 10 to the power 3 to 10 to the power 6 meter square per centimeter cube for a ready microbial growth on the solid gas interface. So there can be this porous solid porous material. It has to be porous, right? So that there is this large surface area, right? Volume compared, there has to be large surface area so that there are large space for that oxygen exchange गर्नको लागि, carbon dioxide exchange गर्नको लागि चाहिँ there should be a large surface area compared to its volume. The matrix should absorb water amounting to one or the several times its dry weight with a relatively high water activity on solid gas interface in order to allow for the high rate of biological chemical activity. So the matrix it has to have the water absorbing capacity high, otherwise that would limit the biochemical activity. So biochemical activity it has to be maximum, and for that water absorbing capacity to material the solid material it has to be maximum. The air mixture of oxygen with other gases and aerosols should flow under relatively low pressure and mix with the fermenting mass. So you put it low pressure, relatively low pressure it has to flow. Aspirating that is normally avoided. So we do it in submerged fermentation. We don't do that. So relatively low pressure compared to the submerged fermentation. Low pressure. The solid gas interface should be a good habitat for the fast development of specific cultures of the molds, yeast, or bacteria, either in pure or mixed culture. Similarly, the mechanical properties of the solid matrix should stand compression or gentle stirring as required for a given fermentation process. This requires general small granular or fibrous particles which do not tend to break or stick to each other. So these are some of the few mechanical properties of the solid matrix. It should be able to stand some gentle compression, folding that it should be able to withstand. And the matrix should not be contaminated by the inhibitors of the microbial activities and should be able to absorb or contain available microbial foodstuffs, carbohydrate, nitrogenous sources, and mineral salts. So the solid matrix is also shouldn't contain certain inhibitors, and it should be rich in carbohydrates, nitrogen sources, and mineral salts. So microorganisms used in solid state fermentation. The microbiological component of solid state fermentation can occur as a single pure cultures, mixed identifiable cultures, or totally mixed indigenous microorganisms. Right? There are different combinations of you can add them. They depend upon what kind of product you are preparing. So some of the examples of solid state fermentation processes are tempeh production, right? Tempeh and enzyme production. Is tempeh? Have you heard about tempeh? Enzyme?
Soybean ko.
Yes, soybean ko. यो नेपालमा चाहिँ अब यसको एकदमै क्लोज काउन्टरपार्ट लिँदाखेरि चाहिँ it is the tofu. The tofu नै हो. हैन अब tempeh and enzyme नेपाली कन्टेक्स्टमा counterpart is the tofu, and soybean ले नै बनेको हुन्छ है, and requires Selective growth of the microorganisms such as molds that need low moisture levels to carry out fermentation with the help of extracellular enzymes secreted by the fermenting microorganisms. Right? So, however, bacteria in yeast which require higher moisture content for efficient fermentation can also be used for SSF but with lower yield. So, यहाँ चाहिँ there is this major problem चाहिँ the bacteria र yield को लागि चाहिँ because they require higher moisture content for efficient fermentation. That's why there is still problem. यसमा चाहिँ problem हुनुको कारण चाहिँ त्यो छ. So there is still more research, the researchहरू bioreactor को डिजाइनमा we have to spend some time. So what are the steps involved in solid state fermentation? So the SSF is normally multi-step process involving the following step. So first step के छ भन्दाखेरि you need to pre-treatment of the substrate material गर्नु पर्यो, right, by the mechanical or the chemical means, right, so that यसको चाहिँ जुन availability छ bioavailability of the nutrients, right, that is available to the microorganisms. With that, you have to reduce its size. And for that, you need to pulverize the straw or the shredding the vegetable material to optimize the physical aspect of the process. However, the cost of pre-treatment must be balanced with the eventual product value. So, there is this part, the economics, the economics of the fermentation process. So secondly, so the hydrolysis of the primarily polymeric material, that is polysaccharides and the protein by the microbial strains. The microbials they'll hydrolyze, and utilization of the hydrolysis product. And finally, there is this separation and the purification of the end product. And so the low moisture content of SSF enables a smaller reactor volume for substrate mass and the liquid surface fermentation, and also simplifies the product recovery. So यसमा चाहिँ के छ भन्दाखेरि so reactor volume चाहिँ एकदम थोरै भएको कारणले चाहिँ per unit substrate mass है त reactor volume चाहिँ के छ smaller reactor volume compared to substrate mass भएको कारणले चाहिँ the recovery process is quite simple. That's why you the major temptation के छ भन्दाखेरि solid state fermentation मा जानको लागि चाहिँ this is the major temptation है त reactor small reactor volume हुन्छ compared to the substrate mass. So however, serious problems arise with respect to mixing, heat exchange, oxygen transfer, moisture control, and gradient of pH, nutrient, and product as a consequence of heterogeneity of the culture. So यसको problems हरु पनि धेरै छ है त. Problem चाहिँ के छ भन्दाखेरि mixing गर्न difficult हुन्छ, heat exchange गर्न पनि it is quite difficult, oxygen transfer, moisture control, all these process लाई चाहिँ control गर्न चाहिँ it is quite difficult in solid state fermentation. That's why we opt for submerged fermentation. So the later characteristics of solid state fermentation renders the measurement and control of the above parameters difficult, laborious, and often inaccurate, thereby limiting the industrial potential of this technology. So these are major hindering factors. That's why there is still adoption is difficult industrial scale. There is renewed interest, but it's very difficult. There is very difficult. Due to these problems, the microorganisms that has been selected for the SSF are more tolerant to wide range of cultivation conditions. So, limited industrial applications for mamatra also used by solid state fermentation. So, solid state fermentation has emerged as a potential technology for the production of microbial products such as feed, fuel, food, chemical, industrial chemical, and the pharmaceutical products. It is widely applied to producing several enzymes, organic acid, flavoring compounds, etc., which must be extracted and purified, then used in different products. Its application in bioprocesses, bioleaching, biobenification, bioremediation, biopulping has offered several advantages. Bioleaching, right? Bioleaching भनेको कसरी थाहा छ bioleaching भनेको? So bioleaching is the industrial application of microbes in mining industry. The bioleaching and biobenification, these are the similar kinds of biological transformation that occurs in the mining industry. mining industry, these are more used. Bioleaching and biobenification, and there is bioremediation. Bioremediation is about using microbes to clear the waste. Waste or generated by it can be some radioactive waste, even radioactive waste, chemical waste, or pollutions those pollution, they can be removed with the help of microorganisms, and that is called bioremediation. Biopulping is about in the paper industry. That is a lot of paper industry. It is one of the most polluting industry, right? And the pulp generate lots of chemicals that are used. In biopulping, microbial transformations, they help in this biopulping. So solid state fermentation, it has advantages, and there are industries where solid state fermentation is used. So the main advantage of such methods is that it produces a minimum amount of waste and liquid effluent, thus not a very damaging to the environment. So this is one of the major benefits of the solid state fermentation. Solid substrate fermentation employs simple natural solids as the medium, right? You benefit. So low technology, so not you don't need a bioreactor, big vessel, right? So you don't need a low energy expenditure, and this requires less capital. Capital it requires less. So there is this advantages of solid state fermentation. No elaborate methods for sterilization, less microbial contamination, and easy downstreaming processes. These are some of the properties of the solid state fermentation. So utilization of agro-agricultural residue as substrate in solid state fermentation processes provides an alternative avenue and value addition to these otherwise under or non-utilized residues. So non or underutilized residues, right? Right, all these residues that are underutilized or not utilized, these residues, they can be used. So the yield of the product is reasonably high. Product yield is reasonably high. So bioreactor design, aeration process, and effluent treatments are quite simple, and many domestic industrial and agricultural waste can be fruitfully used in SSF. And these are some of the benefits, advantages of the solid-state fermentation. What are the limitations? Microorganisms can tolerate only low moisture content can be used. So this is the major limitation. Precise monitoring of the oxygen, carbon dioxide level, and the moisture content is not possible. Solid, it is not possible. Liquid, there is this aeration, right? Agitation, or the impeller, let's say, rotate, you can measure precisely. You can measure oxygen, pH, the O2 levels, and all those processes that are not. All these precise monitoring is not possible in solid state fermentation. The organisms grow slowly, and consequently there is limitation in the product formation. So it takes time. With solid state fermentation, it takes quite a bit of time compared to the submerged fermentation, and heat production, if heat is produced, is very difficult to regulate the growth environment. So compared to submerged fermentation, it's quite difficult with the solid state fermentation. And now we study about the submerged fermentation, submerged liquid fermentation, the kind of fermentation that you are quite comfortable. So fermentation is the technique of using biological conversion of the complex substrate into simple compounds by various microorganisms such as bacteria and fungi. So in course of this metabolic breakdown, they also release several additional compounds apart from the usual product of the fermentation, such as carbon dioxide and alcohol. These additional compounds are called secondary metabolites. Right? So secondary metabolites range from several antibiotics to peptides, enzymes, and growth factors. Right? Example question: what are secondary metabolites? Right? So you have to be able to answer this. The development of the techniques such as solid state fermentation and submerged fermentation has led to industrial level production of these bioreactive compounds. So this is the classical example of the submerged fermentation. Right? So there is this media preparation. Media preparation there is this media sterilization. Media sterilization you pour all those media into this tank, right? Bioreactor. So अर्को स्टेपमा के हुन्छ, there is this inoculum development, right? अर्को चाहिँ एकदमै सानो स्केलमा, लिमिट सानो स्केलमा there is this inoculum development, and you mix the media and the inoculum, right, into this tank, and there is this and then there is this control with all these pH, oxygen, temperature, all these they are controlled by the help of a computer. It can be mechanically control automatically control, and then this finally there's cell recovery, product recovery, and the waste treatment and disposal. So these constitute the submerged fermentation. So submerged fermentation is the method of manufacturing biomolecules in which enzymes and other reactive compounds are submerged in liquid such as alcohol, oil, or other liquid broth. Right? यसमा चाहिँ के हुन्छ, enzymes र bioreactors they are generated, they are manufactured, but they are done in the liquids, right? Either in the nutrient broth or oil or alcohol. It can be in variety of the liquid medium. So we call it submerged fermentation or the liquid fermentation. So there is this free-flowing liquid substrate such as molasses and the broths. So a defining feature here, there is this free-flowing liquid substrate such as molasses and broth, and this process is used for a variety of purposes, mostly in the industrial manufacturing. So what are the principles of the submerged fermentation? Submerged fermentation principle here. Submerged fermentation involves the growth of microorganisms as a suspension in the liquid medium. So these microorganisms, they are in the suspension state in the liquid medium, in which various nutrients are either dissolved or suspended as the particulate solids in many commercial medium, right? These microorganisms, they are in suspension state. To microbes, these microbes, they use this medium, right, nutrient medium, and then they grow and they produce primary metabolites, right, and they also generate the secondary metabolite, the desired secondary metabolite. The submerged fermentation is a process involving the development of microorganisms in a liquid broth, right? So biotransformation ke bhayirako huncha suspension ma or in the liquid condition ma. This liquid broth contains nutrients and it results in the production of the industrial enzymes, antibiotics, or other products. So the variety of products, it can be generated or produced in the liquid broth or in the submerged fermentation. The process involves taking a specific microorganism such as fungi and placing it on a small closed flask containing the rich nutrient broth. So high volume of oxygen is also required for this process. That's why there is this agitation with the aeration process or agitation process. It is mostly used. So oxygen required, meaning there is lots of biochemical activity that is undergoing, and that generates lots and lots of heat. That's why the setup must be quite elaborate. The vessel say elaborate. So bioreactive compounds are secreted into the fermentation broth. Extracellular product of interest, if it is extracellular, it is directly secreted into the fermentation broth. So what is the method of carrying out the submerged fermentation? There are the two common methods by which submerged fermentation takes place. They are the batch-fed fermentation and the continuous fermentation, right? So there is this batch process and there is the continuous process. So in the batch-fed fermentation, sterilized growth nutrient are added to the culture. It is the most common in bioindustries as it occurs during the growth. The biomass is in the fermenter. So it helps raise the cell density in the bioreactor. Cell density inoculum, we have added a very little inoculum into nutrient media consumption bioreactor. So cell density it increases, right? And it's typically highly concentrated to stop dilution. The rate of growth in the culture is maintained by adding nutrient, and also it reduces the risk of overflow metabolism. So there is always this risk of overflow metabolism. Overflow metabolism sometimes for example, alcohol production is the overflow metabolism. The overflow metabolism microorganisms the desired chemical reaction or chemical reaction. That is easy. For example, if you are growing yeast in a very nutrient rich medium, यसले चाहिँ के गर्छ भन्दाखेरि एनारोबिक रेस्पिरेसन गर्न थाल्छ right even if you want it to continue with the aerobic respiration so यसले oxygen use गर्दैन right it doesn't want to use oxygen because since there's lots and lots of nutrient media त्यस्तो कन्डिसनमा चाहिँ ओभरफ्लो मेटाबोलिजममा जान्छ so there is you shouldn't add lots and lots of nutrient media in the bioreactor यसले प्रब्लम क्रिएट गर्छ किन भन्दाखेरि there is this risk of over metabolism है त मैले क्लासिकल एक्जाम्पल भने ओभरफ्लो मेटाबोलिजमको द इस्ट प्रोड्युसिङ द अल्कोहल है त्यो अल्कोहल प्रोडक्सन धेरै न्युट्रिएन्ट रिच मिडियामा हाल्दियो भने चाहिँ इस्ट इट डजन्ट proceed with the aerobic respiration but it proceeds with the anaerobic respiration. An open system is constructed for the continuous fermentation right continuous fermentation you have open system there is this closed system right the batch fermentation there is closed system for majority of the time but in the open system there is this continuous system there is this open system right the mass transfer energy transfer then the sterilized liquid nutrients are slowly and continuously added to the bioreactor at the same rate at which the converted nutrient solution is being recovered from the system. This results in a steady rate production of the fermentation broth. So, there is this achievement of the steady state. The steady state, there is this addition of the nutrient media and there is this removal of the broth from the system. In order to maintain a successful fermentation, certain variables must be monitored. So, एकदमै commonly monitored variables these are temperature, pH, oxygen levels, carbon dioxide levels, and other many levels. Right? Depend glucose level or the alcohol level or it can be certain protein level amino acid level. There are many things to many variables to control and monitor. So some common substrates used in submerged fermentations are the soluble sugars, molasses, liquid media, vegetable fruits and vegetable juices, and sewage and wastewater. They can be used in submerged fermentation. So the applications are mostly extraction of the secondary metabolite that need to be in the liquid form. So some secondary metabolites they have to be in the liquid form, and if that's the case, we can use the submerged fermentation. And it is also used traditionally for the production of the microbially derived enzymes. So like we used submerged fermentation. The industry, by industry, make them commonly used in submerged fermentation. So what are some of the advantages of the submerged fermentation? So advantage is short period, low cost, and high yield. Purification of the product is easier, and in liquid culture, the control of the fermentation is simpler, and consequently, significant reduction in fermentation times can be achieved. So these are some of the advantages. And similarly, the use of submerged culture can benefit the production of many secondary metabolites and decrease the production cost by reducing the labor involved with the solid state fermentation. Submerged fermentation added benefit it can be mechanized and automation is quite easy compared to the solid state fermentation. That's why there is this great cost reduction on submerged fermentation. So what are some of the limitations? So many researchers have demonstrated that solid state fermentation has large impact on productivity, leading to higher yields and improved product characteristics compared to submerged fermentation. The submerged fermentation, since there is this aqueous medium agitation भइरहेको हुन्छ, इम्पेलरलाई मुभ गरिरहेको हुन्छ, that's why the product को जुन purity छ वा product को bioactivity छ, it can get destroyed. है त you are to production गर्दाखेरि चाहिँ के हुन्छ, it can be disturbed. That's why there is always so compared to SSF को चाहिँ के हुन्छ भन्दाखेरि there is this एकदमै high grade को चाहिँ material can be obtained compared to the submerged fermentation. Submerged fermentation मा के छ, there is this risk of impurity हुने chance चाहिँ धेरै हुन्छ. So low volumetric productivity. So volumetric productivity एकदम low हुन्छ. बट हाउ एक्चुअली यसलाई कसरी चाहिँ गरिन्छ नलिफाई गरिन्छ वा यसलाई चाहिँ नेग्लिजिबल गरिन्छ भन्दाखेरि वी जेनेरेट वी समर्स फर्मेन्टेसनमा धेरैभन्दा धेरै भोल्युमहरू जेनेरेट गरेर टु लोवर दिस नम्बर है त So relatively lower concentration of the products and more effluent generation, waste generation पनि धेरै हुन्छ and complex fermentation equipments, right? यो चाहिँ हाम्रो solid state fermentation को भयो, right? So now we'll study about the scale up of the fermentation. The scale up को बारेमा we'll have some study. So do you have any idea about the scale up? Scale up को बारेमा के थाहा छ?
ल्याबबाट इन्डस्ट्रियल स्केलमा प्रोड्युस गर्नको लागि स्केल अप गर्नुपर्छ पहिला पाइलट स्टडी गर्नु पर्यो पाइलट स्केल
So what happens? Okay, thank you. What happens if we don't conduct scale up studies? What are some of the challenges we might face if we don't conduct scale up studies? के हुन्छ होला किन गर्ने त अब के छ उसको नगर्दै किन गर्दाखेरि हुँदैन के हुन्छ उसको घाटा के हुन्छ होला के त छ होला घाटा
फिजिबल छ कि नै भनेर चेक गर्न पनि हो
होइन मैले भन्न खोजेको फिजिबल छ कि छैन भने त ल्याब स्केलमा थाहा पाइहालिन्छ नि इन्जाइम प्रोडक्सन गर्छ कि गर्दैन त्यो ब्याक्टेरियाले यु क्यान डिटरमाइन यु क्यान आइडेन्टिफाई द्याट इन द ल्याबमा यता ल्याब स्केलमा प्रोडक्सन गर्दाखेरि नै दिस माइक्रोब यु हेभ सर्टेन आइडिया द्याट दिस माइक्रोब विल जेनेरेट अमाइलेज हाई अमाउन्टमा अमाइलेज यसले चाहिँ हुन्छ नि जेनेरेट गरिरहेको छ एकदमै भनेर चाहिँ यु हेभ द्याट नलेज सो वाइ कन्डक्ट स्केल अफ स्टडिज सिधै ल्याब स्केलबाट चाहिँ इन्डस्ट्रियल स्केलमा गयो हुँदैन वाइ गो थ्रु दिस पाइलट स्केल अफ what is the rationale behind going through this pilot scaling up garnako चाहिँ what is the rationale के छ होला rationale any idea
कहिलेकाहीँ हैन थोरै इन्क्रिज गर्दा फेरि इनपुटमा लाइक अब प्रेसर इन्क्रिज गर्यो भने चाहिँ अनि पछि आउटपुटमा धेरै इफेक्ट आउने हुन्छ त्यस कारणले त्यो पनि थाहा पाउनको लागि के होस् इफेक्ट
okay है त let's study है त scale up why we should do why it's very essential है त why it is absolutely essential you go through the scale up studies and if you don't go through the scale up studies and you are directly moving, you are directly jumping from the lab scale to the industrial scale, you are bound to fail. Absolutely bound to fail. So I'm going to study about the scale up properties. So scale up studies refers to the act of using results obtained from the laboratory studies for designing a prototype and a pilot plant process, construction of the pilot plant and using pilot plant data for designing and constructing a full-scale plant or modifying an existing plant. So this refers to the scale-up studies. So what is the deal? First, you do the basic research or the fundamental research in the laboratory. So for example, you want a microorganism that generates lots and lots of amylase, enzyme amylase, and what you do that is you go in the nature out there and you select some certain strain. The certain strain select you have identified that at least in the lab setting it can lab setting petri plate petri plate ma beaker ma 1 liter ko beaker ma right small petri plate ma or 1 liter ko 2 liter ko petri plate ma sorry jar ma that it is able to produce the desired amount of amylase right but say you want to scale that process to the 1000 liter plant right what happens is that to 500 or 1000 liter vessel ma before growing the microorganism, the desired microorganism to produce the enzyme amylase, what you do is that you do the experiment in 20 liter or 30 or the 50 liter. 50 liter ma चाहिँ you do the designing है त कसरी गर्ने, what are the different variables. के हुन्छ भन्दाखेरि there are certain variables that is only present in the when the quantities are in large component है त, right? जस्तै water, if you take a single drop of water, a single drop of water लियो भने that doesn't have the property of taste. So taste हुँदैन. It doesn't have the property of taste, it doesn't have the property of color, it doesn't have the property of viscosity. So many different properties are not present. एकदमै धेरै few amount मा 5 वटा वा 10 वटा hydrogen water molecules if you gather, it doesn't have certain those kind of properties. Similarly, के छ भन्दाखेरि 1 लिटरमा गर्दाखेरि र 1000 लिटरमा गर्दाखेरि के हुन्छ भन्दाखेरि there are many properties that are not observed when you are conducting studies in the lesser volume. Property नै हुँदैन, right? Suppose there is no property of थोरै स्केलमा जेनेरेट गर्दाखेरि maybe the carbon dioxide generated, it will not lead to the increase in the temperature of the vessel because के छ भन्दाखेरि volume थोरै छ, surface area धेरै छ, right? So के छ भन्दाखेरि that's why there is never the problem of the heat exchange. तर धेरैमा चाहिँ if you are generating, right, धेरैमा जेनेरेट गर्दाखेरि चाहिँ there is this problem of heat generation. For example, अब के हुन सक्छ भन्दाखेरि कुनै एउटा चाहिँ प्रोसेस चाहिँ एक्जोथर्मिक छ. there is a process that is an exothermic process. थोरै भोल्युममा गर्दाखेरि चाहिँ it won't matter के, किन भन्दाखेरि थोरै भोल्युम छ, सर्फेस एरिया अटोमेटिकली धेरै भइदिन्छ, थोरै भोल्युम हुँदा सर्फेस एरिया धेरै भइदिन्छ. That's why to heat exchange say it is never the problem. अब 1000 लिटरमा गर्दाखेरि चाहिँ के छ भन्दाखेरि भोल्युम धेरै छ, सर्फेस एरिया एकदमै लिमिटेड छ, त्यो भएपछि there is this problem of heat generation. Heat generation मात्र होइन, there is the problem of viscosity, there is a problem of differential, the differential temperature को gradient बनिन्छ, pH को gradient बनिन्छ. All these properties हरु चाहिँ these are not seen in low volume है त. 1 लिटरको भोल्युममा there is very little chance of forming a pH gradient त्यो liquid medium मै. तर 1000 लिटरमा चाहिँ there is chance of a pH gradient भित्रै त्यो vessel भित्रै there is this formation of a pH gradient. एउटा सेन्टर तिर चाहिँ pH 5 छ अनि त्यसपछि vessel नजिकैको रिजनमा चाहिँ there is pH 8. So there is this pH gradient and these properties are not seen, right, in lower volume. Lower volume we don't see these properties. त्यसलाई हामी emergent properties हरु पनि भन्छौं. When एकदमै you say these are emergent properties, and these properties are only seen when there is maximum volume. Number चाहिँ increase हुँदाखेरि देखिने properties हरु. मैले भने नि जस्तै एउटा सिंगल मोलिक्युल अफ वाटर को कलर हुँदैन, there is no color. कलर हुने जेनेरेट हुने भनेको अलिकति नम्बरहरु बढ्दै गएपछि जेनेरेट हुने हो. And similarly यो अरु properties हरु पनि they are not seen. अब कुनै property observe गरिएको छैन भने how can you design the system? Lab scale मा त property देखिँदैन भने how will you design that in an industrial scale? That's why a pilot scale is needed. So laboratory scale, there is this pilot scale, and then there is this industrial scale, and there is this increase in the power and the magnitude. And this we call the scale-up studies. A pilot plant allows the investigation of the product and the process on the intermediate scale before large amounts of money and resources are committed to the full production, right? So you know that the microbe is able to generate amylase in the lab scale, but it might not be the case. धेरै भोल्युममा हुँदाखेरि, धेरै नम्बरमा हुँदाखेरि maybe the bacteria doesn't feel like producing the amylase. Certain volume भएपछि, certain number भएपछि, maybe the bacteria doesn't like to produce in higher volume. Maybe there is this relationship between volume of the bacteria, right, and the amylase production. All these factors, it can play a very important role, and these factors many times they are not observed. So it is not usually not possible to predict the effect of many-fold increase in the scale. So many times it is not possible to predict either. 5जना मान्छेलाई चाहिँ खाना पकाउनु र 5जना मान्छेलाई खाना पकाउनु इज अ डिफरेन्ट league हो. It's a separate league. So similarly is the case for the pilot studies. So scale-up studies are the studies carried out in the laboratory or even pilot scale fermenters to yield data that could be used to extrapolate and build the large-scale fermenters with sufficient confidence that it will function properly with all its behavior anticipated. So nature, obviously, you would like to anticipate the behaviors in the large scale. So the importance of the scale-up studies are that more important during scale-up exercise is that we are trying to build industrial-size fermenters capable or close of producing the fermentation product as efficient as those producing the small-scale fermenters. So this is the importance. It must be appreciated as the size of the fermenter increases. Just during scale-up properties, various parameters might not show a predictable linear correlationship. So linear correlationship no such. So you may have predicted that if you increase the volume, right? If you increase the volume, temperature of the medium temperature increase. And you might have predicted, but maybe there is this non-linear relationship. Right? हाम्रो assumption हुन्छ क्या हाम्रो ह्युमन माइन्डको के हुन्छ भन्दाखेरि र बायसनेस के हुन्छ भन्दाखेरि देयर इज दिस लिनियर रिलेसनशिप हुन्छ भनेर 5 men can do a job in 10 days. How many men can 1000 men do the job भन्ने जस्तै. So 1000 men हुन थालेपछि there is this collision हुन थाल्छ, there is this gang बन्न थाल्छ, and then they'll start quarreling with one another. That's why what happens is that maybe 5 men will do the job earlier than the 1000 men. So that because these effects are not quite linear, right? It's a linear हुँदैन. So similarly त्यस्तै किसिमको fermentation मा पनि or the bioreactor मा पनि this can be observed. Certain parameters change, certain remain constant. Some parameters need to be modified and adjusted during the scale-up studies, right? That there are these parameters like your pH, temperature, all these are parameters. Parameters पनि भन्छ like variables पनि भन्छ है त. So objective is to try to get the same fermentation efficiency as obtained in the small scale fermenters at the most economic values. So these are the importance of the scale-up industries. So there are some crucial studies which will only be answered by carrying out a pilot plant, such as determining the various operational parameters for optimized oxygen supply to the fermentation process, right? What is oxygen demand ko barema, selection of the optimum operating modes of the fermenter, kun mode ba run गर्ने, right? So determining the changes in the rheological properties and its effect on the fermentation process, right? Temperature increase हुँदाखेरि they change in the rheological properties. Rheological properties भनेको for example viscosity, say it comes under the rheological property, right? So what happens if there is this increase in the temperature, right? Or 1 liter मा त you could have predicted, right? So 1000 liter मा त it is very difficult to predict. So for that prediction गर्न सकिदैन. So prediction गर्न नसकेको कारण you have to do the experiments, है त. सबै एक्सपेरिमेन्टहरु प्रेडिक्ट बायोलोजीको एउटा एकदमै बायोलोजीमा हामी why do we do so many experiments in biology? किन होला? Anybody? कसलाई थाहा छ? Why we do so many experiments in biology? किन biology is an empirical science? Physics को relationship मा त biology is a very empirical science.
Evidence based हुने भएकोले होला.
Evidence based त हजुर evidence based सबै science evidence based हो नि. Evidence त दिनै पर्यो नि.
पछि अल्टिमेटली अब ह्युमनमा युज हुने हो त्यसको लागि टेस्ट आउट गर्नुपर्छ नि त अरूमा.
बायोलोजिकल सिस्टम कम्प्लेक्स भएर होला त्यो इन्टरेक्सनहरुले गर्दा छुट्टै इफेक्ट हुन सक्छ.
Biological systems are so complicated you cannot predict. Predict गर्न सकिदैन. You cannot beforehand prediction गर्न सकिदैन. That's why predict गर्न सकिन्छ, right? सकिन्छ, but the prediction will be absolutely wrong prediction. सो के रहेछ भन्दाखेरि मेन भनेको चाहिँ इट्स बिकज अफ द कम्प्लेक्सिटी है त कम्प्लेक्सिटी धेरै छ यसमा चाहिँ कियोसको इलिमेन्ट धेरै छ है त अहिले आएको छ नयाँ न्यू साइन्स साइन्स अफ कियोस भनेर द्याट्स वाइ देयर इज इभन इफ देयर इज अ स्लाइट डिस्टर्बेन्स स्लाइट डिस्टर्बेन्सले के गर्न सक्छ भन्दा इट क्यान क्रिएट है त एकदमै लार्ज स्केल पर्टुबेसन्सहरू लिएर आउन सक्छ द्याट्स वाइ अब त्यो गर्नको कारण प्रिडिक्सन गर्ने टुल्सहरू मोडल्सहरू चाहिँ दे विल फ्लप है त त्यो फ्लप भइहाल्छ द्याट्स वाइ अब त्यसको लागि चाहिँ द बेस्ट वे इज टु डु द एक्सपेरिमेन्ट गरेर आफै पत्ता लगाउनु नै हो द्याट्स वाइ स्केल अफ स्टडिजमा पनि वी हेभ टु कन्डक्ट दिस स्केल अफ स्टडिज बिकज वी क्यानट प्रेडिक्ट देम प्रेडिक्टै गर्न सकिँदैन द्याट्स वाइ बेटर डु द एक्सपेरिमेन्ट्स एन्ड डिटरमाइन द के छ अक्सिजन सप्लाई कति चाहिन्छ भने डिटरमाइन गर्ने है कुन मोडमा चलाउने है त कति डिफ्रेन्ट है त इम्पेलरको भेलोसिटी इम्पेलरको स्पिड टेम्परेचर त्यो कोटको ज्याकेट टेम्परेचर अल द्याट कुन कुन मोडमा रन गर्ने अर द रियोलोजिकल प्रपर्टिजहरू भिस्कोसिटीहरू चाहिँ हाउ दे आर चेन्जिङ है त ओभर टाइम है त डिफ्रेन्ट डेमा डिफ्रेन्ट टेम्परेचर डिफ्रेन्ट पिएच कसरी भेराइ भइरहेको छ एन्ड जस्ट ट्राई टु एन्ड देन ट्राई टु कम अप विथ अ न्यू मोडल है त फर्मुलेसन मोडलिङ एन्ड फर्मुलेसन अफ द कन्ट्रोल प्रोसेस एन्ड सेन्सर्स एन्ड कन्ट्रोल्स सो त्यसपछि चाहिँ युट्युब अब त्यसपछि के गरिन्छ भन्दाखेरि ल्याब स्केलकोले चाहिँ इट वन्ट वर्क सो पाइलट स्केलबाट चाहिँ यु विल ट्राई टु जेनेरेट अ मोडल so that will be a little better model than the one that you can generate from the lab studies. So what are some of the steps in the scale of proper steps? So define product economics based on the projected market size and competitive selling and provide guidelines for allowable manufacturing cost. Conduct laboratory studies and scale up planning at the same time. Define key rate controlling steps the proposed process. Conduct preliminary larger than laboratory studies with equipments to be used in rate controlling step to aid in plant design. And this like you have to determine the rate controlling steps also whether that you have to be defined. You use steps rate controlling steps also beforehand you have to be able to come up with a certain idea है त यहाँनिर चाहिँ टाइम बढी लाग्न सक्छ यहाँनिर चाहिँ 5 दिन लाग्छ है त यो स्टेपमा चाहिँ इट विल टेक 5 days है त ओके यो स्टेपमा चाहिँ यु हेभ टु बी लिटल बिट मोर कसियस बिकज यसमा चाहिँ प्रोडक्ट डिग्रेडेसन हुन सक्छ अल द्याट थिङ यु हेभ टु डिजाइन है त So design and construct pilot plant including provisions for process of environmental control, cleaning and sanitizing systems, packaging and waste handling systems, and meeting regulatory agency requirements. सबै कुराहरुलाई चाहिँ you have to be you have to take care. कसरी यसलाई चाहिँ क्लिनिङ गर्ने कसरी चाहिँ how you manage the waste material, how you allow regulatory bodies or requirements or you can check cleaning, sanitizing, packaging, all that thing. Laboratory scale ma you don't do. Laboratory scale ma ke garincha you just isolate an organism and त्यसपछि spectrophotometer मा गएर you just check यसले यति चाहिँ amylase generate गरेछ यसले यति कम amylase generate गरिरहेको छ भनेर. But in the industrial scale up चाहिँ के हुन्छ भन्दाखेरि so many कुराहरु चाहिँ sanitization problem हुन्छ. You don't have the sanitation problem in laboratory studies. Laboratory studies matter there is no problem of sepsis. You know, lab good ma garira huncha काम, right? Laminar flow ma. That's why. So laboratory scale ma there are very limited problems, but scale up studies ma you have to try to include all those problems that you might encounter in the industrial scale. Our sanitation, cleaning, like the different processes or rate limiting steps or so evaluate the pilot plant results, product and processes, including process economics to make any corrections and a decision on whether or not proceed with the full scale plant development. About full scale plant development. That the plant elaborate plant you have to come up with the final answers. Scale up industry. So the most scale up studies are usually carried out at different phases involving different scales of the fermenter. So you can go with the different data. अब आज चाहिँ किन भन्दाखेरि आज चाहिँ laboratory scale गर्यो त्यसपछि 50 liter त्यसपछि 100 liter त्यसपछि 500 liter and then you can go to the 1000 liter. Serially you can go with that. Preliminary work are carried out at different levels of petri plate and small scale laboratory fermenters to establish whether the process is technically viable, meaning it is possible to produce such fermentation process and the products on the small scale. Small scale ma, laboratory scale ma, you have to confirm your idea is actually viable or not. Viable छ कि छैन, lab scale में viable छैन भन्दा it won't be viable at the industrial scale. No need to go to that all that trouble. You just stop at that process. And if it's viable at the laboratory scale, you undergo the different pilot scale up projects. Additional parameters not provided by the Petri dishes studies and far more confidence are required by carrying further studies using submerged liquid fermentation using various sizes, laboratory scale fermenters, and even the pilot plant fermenters. So it's a different level. My industry, you can develop the confidence. Right? Okay, आज हामी यति पढौँ है त नेक्स्ट क्लासमा हामी will wrap up this chapter. है त Vijay, do you have any questions and queries?